I'm going to talk about part of my dissertation research on lionfish-associated bacterial communities, specifically the surface bacterial communities, and their possible role in pathogen inhibition. So as we know, lionfish is a beautiful Indo-Pacific fish, which is highly invasive in the Atlantic, Caribbean, and Gulf of Mexico. Introduced about 30 years ago, it has spread and inundated most of our tropical shorelines here. So whether they're terrestrial or aquatic, invasive species tend to share 7 common attributes. When my work began, 5 of these 7 were already known for the lionfish. 1, similar environmental conditions between the native Indo-Pacific and the invaded Atlantic allow the fish to stay within their physiological limits. 2 is the absence of powerful predators. Lionfish have these 18 highly venomous spines, so they're not very palatable. So there's no consistent predation known in the Caribbean on lionfish. Number 3 is exhibiting unique behaviors. Lionfish use hunting techniques that have never before been experienced by native Atlantic fishes. So they can actually herd prey and eat them by rapid strike. Number 4 is being a habitat generalist. Lionfish can live pretty much anywhere that there's saltwater, substrate, and food, like in this mangrove habitat, which is particularly worrisome because these are important nursery grounds for coral reef-associated organisms. And 5 is an increase in mass. The lionfish on your left was collected in Belize. Lionfish on the right was collected in Taiwan. You can use my hand as a scale in both of those images and see how much bigger the actual lionfish are here in the Atlantic. In fact, they're becoming so fat that they're now exhibiting signs of obesity. Welcome to America. The final 2 attributes, pathogen release and symbiont retention, were not known when my research began. So today I'm going to talk about some of the symbiont retention work that I did. So I've studied the surface-associated bacterial communities. And in a biogeographical study of lionfish across both their ranges, I found that the bacterial communities of lionfish are not significantly different between the native range and the invaded range. But interestingly, the bacteria plankton, so the bacteria in the water column between those 2 ranges, were significantly different. So this indicates that somehow lionfish may be differentially selecting the bacteria from their environment. So for this work, I wanted to look at what's the point of that retention? Why do we have that poor bacterial community? So in other studies of surface-associated fish surface-associated bacterial communities, we found that those bacteria have a wide spectrum of possible benefits. So in these large swimming pelagic fish, bacteria can actually help in drag reduction, so there's efficient locomotion of the fish. Also pathogen inhibitions, this is seen on this plate, and I'll explain what that's showing shortly. And not only can they inhibit the growth of pathogens, but they can also block the adherence of pathogens to the surface at all. So not only do they stop the growing, but the pathogens can't even adhere to the fish. Unfortunately, this fish was not able to do that. So the objectives of this study were to determine if the lionfish surface-associated bacteria were able to inhibit pathogens, and if that ability was seen in both the native and invaded range, and if the strength of that inhibition between the ranges was similar. So this study consisted of a lot of little assays, like most microbiology studies do. So I'm going to present each one with its results and bring you back to this flowchart to keep everything organized. So lionfish were collected from the native and invaded ranges. Their surfaces swabbed and the bacteria isolated. The native range included collection from Taiwan, Central Philippines, and Indonesia. And the invaded range were Key Largo, Bahamas, Belize, and Honduras. So 149 isolates were grown, and they were all tested for antibacterial activity by disc diffusion assays. So each isolate was grown in a liquid culture for 14 days. The culture was then spun in a centrifuge to pellet out the cells, and what you're left with is the cell-free supernatant. So in order for the antibacterial activity of the bacteria to be helpful for the fish at all, it has to leave the bacterial cell. So we don't want any growth of the isolate cells to be muddying up our results. We want it in the cell-free supernatant. So we separate that out, and we made these seeded plates with a standardized amount of known fish pathogens. So all 149 isolates were tested against 6 strains of known fish pathogens. So the cell-free supernatant was seeded or infused into these filter paper discs. The filter paper discs then put on the plate, and the plate grown overnight. So you get a lawn of pathogens, and where you see these zones around the discs indicate pathogen inhibition. The disc in the top right corner was soaked in gentamicin, so you see a very large zone. That's a known concentrated antibiotic. And you see discs from 3 lionfish isolates with obvious zones of inhibition, and 2 which did nothing at all. So of the 149 isolates from both ranges, 54 were able to inhibit the growth of pathogens. 21, or nearly half, of the invaded isolates had this ability, and 33, or nearly a third, from the invaded range had this ability. So this is very qualitative, so we wanted to get some measurements of this strength. And so all of the isolates which inhibited at least 2 pathogens went into minimum inhibitory concentration assays. So I used the techniques of these assays, but did not necessarily determine the minimum inhibitory concentration. You'll see why shortly. But this assay gives us an idea of the strength of pathogen inhibition and growth, percent growth inhibition of the pathogen, as well as the ability to dilute the metabolite. So what happens is I prepared the cell-free supernatant as before, but instead of infusing a filter paper disc, I serially diluted it in sterile marine broth. Marine broth. So I ended up with 100%, 50%, 25%, and 12.5% of that cell-free supernatant in a solution. I added cells of Vibrio parahemolyticus to that solution, so it's a common fish pathogen, and let them grow overnight. Then measured the spectral absorbance at 600 nanometers, and this was used as a proxy for the number of cells of that Vibrio, that pathogen, that was able to grow in the cell-free supernatant of these isolates. So I have this broken up with the isolates from the native range and isolates from the invaded range. On the y-axis, you have percent pathogen inhibition. X-axis is your isolate. And each of the groupings, starting from your left, goes from 100% and then ends at that 12.5% cell-free supernatant. And so we saw a lot of variability in both the strength of pathogen inhibition and the ability to dilute that metabolite out. So I'm going to point out some of the more interesting patterns we saw. So here in this isolate, at 100% cell-free supernatant, there's 90% pathogen inhibition. But it was very easily diluted. And by the time we got to 12.5% cell-free supernatant, there was no inhibitory effect. This was the native isolates. Here for the invaded range isolates, we see similar variability in both strength and ability to dilute. Another interesting pattern here, where at 100%, it's not quite as strong. We're between 70 and 80% pathogen inhibition, but we could not dilute that strength out. So even at 12.5%, we still had 70% pathogen inhibition. Overall, based on analysis of variance, the effect of dilution was significant, but there was no significant difference between the strength of pathogen inhibition between the native range and the invaded range. So we're seeing similar pathogen inhibition, at least potentially between both ranges. We still don't know who's responsible for this. So is this a part of that core bacterial community, or is it just common on the ability common? And so I went back to those 54 isolates that were able to inhibit a pathogen, extracted DNA, amplified the 16S rRNA gene, so an identifier for bacteria. Of those 54 isolates, there was still potential that we had multiple isolates of the same species. So in order to be economical, I tested, so I didn't want to sequence the same ones over and over. So what I did is I took the 16S gene, digested with a restriction enzyme. The restriction enzyme looks for a specific sequence pattern of the gene and cuts it. So you get different fragments based on the sequence of that gene. So really closely related isolates are going to have similar sequences, those show similar banding patterns. So digestions were run out on a gel. Isolates with similar banding patterns were grouped into taxonomic groups, and a representative from each group is then sequenced. So here's an example of one of those groups. It just so happened to be nice and next to each other. So one isolate of those got sequenced. They weren't all that easy. Some of you had to go, I had lots of these images, had to go between. We have a computer program that helps though. So what I found is there was a lot of diversity in the bacterial isolates that were able to inhibit pathogen growth. And that this diversity was similar between the isolates from the native range and the invaded range. So the native range are indicated with the yellow star, the invaded range with the purple box, and so you can see that they are pretty evenly dispersed throughout this tree. This group were the gamma proteobacteria. These are really common in the marine environment. And in previous work I've done on just metagenomics of the lionfish bacterial community, we were very dominated by these gamma proteobacteria. Interestingly, we saw Pseudomonas species from both ranges, and these are known to be bioactive bacteria in the marine environment. We also found members of the Bacillus family and genus. Bacillus are important gram-positive bacteria, which are known to be bioactive and have also been known to common in host-microbe interactions for pathogen resistance. Here we do see some grouping between those from the invaded range and those from the invaded native range. So there's some interesting patterns that might be more of an environmental effect. So my work has shown that the lionfish core bacterial community is potentially beneficial to the lionfish in pathogen inhibition, and that there's similar diversity and strength of that inhibition between both ranges. And so this indicates these members that are able to inhibit pathogens are likely part of that core bacterial community. So it appears that lionfish have retained symbionts, which is an important contributor to the invasive ability of lionfish in the invaded range. So I'd like to thank people that let me use their lab that have speared lionfish with me, and my resources. Oh, yes. So in my first study when I did the metagenomics, I did not find any evidence of disease on lionfish or squirrelfish, so they're common in the Atlantic. But found in the community there were pathogens on the squirrelfish surface, but none on lionfish that I could find. And so it doesn't really indicate much, but there's of what we were looking at, I have not seen a diseased lionfish. Yes?
Have you looked at the surface of other fish, native to Caribbean, to see what's going on?
Yes, so I showed in work I presented before that the bacterial communities of lionfish are significantly different than the bacterial communities of 3 different species of native Caribbean fish. What's really interesting is that